I wanted to make you something that would help you with the macromolecules lab. Just having the pictures doesn't really tell you what we would be doing in the lab. So this little activity duplicates sort of what we would have been doing in the lab. So when you open the activity, you see this. And this is what you would normally see in the lab. You'd see a box of reagents, burette, iodine, Sudan-4, and Benedict's that test for the different macromolecules. And then you would first establish the controls with water, albumin, which is your protein, starch, and corn oil. And the first thing we would give you a bunch of tubes, and we would ask you to put those in a rack. So we're going to do this on the left-hand side here. You see what you're supposed to be doing. That's the list of things to do. So the first thing you want to do is you set up the tubes. To do that, you just click on this button here that says Set Up Tubes. So click. It's going to set up the tubes. Now, I made this thing so it's not, perfection or anything like that, but hopefully it's going to get you close. The next thing you want to do is you want to label the tubes, because you always want to label the tubes because, liquids always look the same and you got to figure out what's what. So label the tubes. And then after you label the tubes, you're going to add the solutions. So here are your solutions: your water, your albumin, your starch, and your corn oil. And over here, this tells you what tube you're going to put what solution in. So your water goes in all the ones that are labeled 1. Your protein goes in all the ones that are labeled 2, etc. So the way you add water to the tube is by clicking where it says Add Water. And then you say which tube you want. A1, if you capitalize it, it's great. It'll also work with lowercase. And then it'll put a bunch of drops into this first tube. Then you're going to click, you're going to click B1, and it's going to put, the dropper is going to put some water into B1. Now, one of the beautiful things that this experiment can do that we can't do in the real lab is if you say, I would like to put some drops into A2, it's going to tell you don't do that. So you should not add water to this tube. When we do this in real life, there's always somebody who puts something in the wrong tube. So you can't mess this one up. Well, ish. You can't mess sort of kind of this one up. So I'm going to pause now. So the first thing you're going to do is you're going to add water, protein, starch, and lipid to the tubes according to this plan. I'm not going to waste my time or your time doing this, and I'm going to now basically do this. I'm going to pause the video while I do this, and then we're going to get back to I want to get back to the video once I have all the solutions added that I need. So I've added all the solutions to tubes A, B, and C. I haven't added anything into D yet. Now, if this dropper here bothers you at the very end, unfortunately, it doesn't go away by itself. So you can click on here to just clean up the dropper, and it'll make the dropper go back to the tube. For part 2, it's a little different. That's where the D tubes come in. And in order to see the parts 2, you can open. So this one here tells you all the solutions have been added. And in order to work with the D tubes, you're going to have to open part 2. But before you do that, that's just the Benedict's reagent. So let's take care of A, B, and C first. So in all the A's, you're now going to add burette. So if you click on that, you then say, I'm going to put this into A1, and it's going to put this into A1. Then you're going to put this into A2, and it's going to put it into A2. And one of the things you notice when you put it into A2, I'm going to put it into A3 too, notice that A2 changed colors. And that's what you would see in real life too. So you added the solutions that were added here is all the 1's got the water, all the 2 got the protein, all the 3's got the starch, and all the 4's got the lipid. And burette tests for protein. So the tube that had the protein in it tested positive for protein. So purple means it's positive for protein. The second thing I want you to see here in the, let's go clean up the dropper again. Well, should probably clean up the drops too, but so the next thing you see is that there's a lipid added here to the bottom, right? That's your lipid control, and lipids don't mix with water, so you end up with two layers here because burette is a non-polar. It's a watery solution. No, it's a polar. It's a watery solution. Lipids are non-polar. And again, I'm going to add all the reagents now. I'm going to pause the video again, add the rest of the reagents, and then we're going to deal with the D tubes. So after adding the iodine to row B and the Sudan 4 to row C, you can see that the iodine, the negative result is yellow, the positive result is black, and note that starch is the one that gives a positive result with iodine because iodine tests for starch. And so that is the only positive. The others don't react positively to that. And then Sudan 4, let's go clean this dropper up again. Sudan 4 reacts with lipid. And the way you see whether this is positive for the lipid is you look for the layers. So the Sudan 4 that we have is a lipid-based solution. So it mixes only with lipids, and Sudan 4, the reagents itself, turns lipids red. So a negative result would be two layers, one red and one whatever the color of the unknown of the solution was. And if you don't get two layers and it's red throughout, that's a positive result. Now to figure out what you're supposed to do with row D, column D that is, you click on here where it says open part 2. So click on that. And that opens a bunch of more solution and another track for you here, another chart here for you. So here in D1, you put water, then 1% glucose, 10% glucose, and 10% sucrose, because the Benedict's tests for reducing sugars. So let's add water to D1, add 1% glucose to D2, 10% glucose to D3, and 10% sucrose to D4. Once you add, and now we're going to add Benedict's to all four of those. And again, I'm going to pause, well, I'm just going to show you. Add Benedict's to D1, add Benedict's to D2, add Benedict's to T3, add Benedict's to D4. And you see no difference between all four of them. Well, that's because the Benedict's needs to be activated in quotation marks. So what you have to do is you have to take these tubes and actually heat them up in a water bath. So we're going to click on here where it says heat the tubes. Well, I'm going to clean up the dropper first because it bothers me. We're going to heat the tubes and ignore the flickering. So when you heat the tubes, they go into the water bath and then they change color in the water bath. And to make sure that you can see what goes with what here, you can then re-rack the tubes. And so these are your controls. So your controls show you that with water, you get negative results. So that's number one. That's anything that has a 1, right? You get negative results for everything. With protein, you get positive for protein with the burette, and you get negative results for all the rest of them. Now the D1 through D4, remember those are different solutions, right? With starch, you get a negative result and a positive result for starch, negative for lipids, and then with lipids, you get a positive result for the lipid. In the for the Benedict's for experiment, you see that the water is negative, the 1% glucose is slightly positive because glucose is a reducing sugar, and a slightly positive reaction with Benedict's is green. It's semi-quantitative, so the 10% glucose gives you an orange reaction, which is the more intense reaction. And I explain in the lecture why the red is more positive than the green. And then the sucrose, you end up, that's a non-reducing sugar, so the sucrose is a negative result. So these are your controls that you're going to now compare your unknowns to. And you've got pictures of all of those, so you can compare and contrast the things you see here with the pictures that are in the lab handout. Now to do the unknowns, then you go and you click on the open worksheets for the unknowns. So click on here, and let's say you want to start with the first unknown. The way I've got this set up is each worksheet actually contains 4 unknowns. So if you say OK, it takes you to the worksheet for the 4 unknowns. And so you do the same thing that you did before. You've got the A, B, C's, and D's. You've got the water here. And then you've got your unknowns 1 through 4. And then you've got your burette, your iodine, your Sudan 4, and your Benedict's. This time around, you have the, you're adding the same thing into column 1 through 4 and A through D. So you're not separating them out into 2 experiments. So set up the tubes, label the tubes, and now you're going to add unknown number 1 into A1, into, click again, B1, C1, and D1. And I'm going to pause this again and add the other reagents to all the others. Not the reagents, the unknowns. Sorry. Once you added all the solution, let me clean up that dropper again because it bugs me. Once you added all the solution, the sentence, all the solution have been added, shows up here. And note that these solutions have a different color. Those colors aren't random, right? So this is milky white on purpose. And then you're going to add the reagents. So you're going to add burette to A1. I'm going to add it to A2. Add it to A3. And you add it to A4. And again, I'm going to pause and add iodine to all the B's, Sudan 4 to all the C's, and Benedict's to all the D's. So you can already tell that unknown sample number 2 seems to have protein in it because it tested positive for protein. So did unknown sample number 3. It looks like none of the samples have any kind of starch in them because none of them turned black. And it looks like sample number C2, sample number 2 is a lipid, or lipid-based anyway. So it's got both protein and lipid in it. Now for the Benedict's test, again, remember we need to heat that one up. So we're going to heat up the tubes, and they change colors, and then we're going to re-rack them. And so what you see here is that sample number 2 seems to have protein in it, no starch. It does contain lipid, and it seems to contain a small amount of sugar. Sample A this result is not light blue. This result is kind of an orangey color, and you'll see the same thing on your picture that you have in your lab handout. That's because that test is very similar to the Benedict's test. And so if there's a lot of reducing sugar in there, this light blue goes into a light orange color, but that is not a positive. That is still a negative. For protein, only purple is positive. So A1 is going to be negative for protein, negative for starch, negative for lipid, and positive for reducing sugars. And then you're going to go through all of these individually, just like that. And then the quiz gives you the ability to guess at what's what. When you're done with these unknowns, you can click on the worksheet, and then you can say, all right, I'd like to start working on samples 5 through 8. And again, you get the same kind of setup. You've got unknowns 5 through 8 here, and you do the same thing. You set up the tubes, you label the tubes, you add the different reagents, and then you go ahead and open the last worksheet, which contains the samples 9 through 12. And this gives you the ability to sort of review, in quotation mark, hands-on, right? More so than we could just by using the pictures. And I hope this helps.